Here in 11.6, we're going to talk about how prokaryotes achieve genetic diversity. So if we think about organisms like ourselves, for example, humans, when we have an egg and a sperm come together, there is genetic diversity because we have meiosis, right? So we have meiosis occurring, and within meiosis we have things like crossing over occurring and independent assortment, and we have various mutations and things. And all of these are happening in both the male and the female gametes. And then when those two gametes come together, they're very different than both parents. They're an organism that has traits from both parents, but is a new unique organism as far as its specific genes are concerned. So when we are talking about prokaryotes, remember they go through binary fission. When they go through binary fission, most often, or we have budding and fragmentation, things like that. But when they go through binary fission, it's an identical copy. So there's not a lot of genetic diversity in just its typical regular binary fission. So prokaryotes have different ways of achieving genetic diversity. So when we were first talking about something like humans, we are talking about vertical gene transfer. So this is when we have the transmission of genetic information from one generation to the next generation. So transmission of genetic information from one generation to the next generation. And this is what we see, again, we see this in humans. And then this is the main mode of transmission in all cells. So main mode transmission in all cells. So this is what we see in sexually reproducing organisms. So as I mentioned, when we are talking about humans, for example, which are sexually reproducing organisms, we see vertical gene transfer. We see vertical gene transfer in asexually reproducing organisms as well, but it's going to be identical. Versus in sexually reproducing, like you just mentioned, we have things like crossing over that's going to adjust the genes. We have independent assortment. If you don't remember what these things are, definitely go back over your meiosis so that you remember what crossing over is and independent assortment. And then we just spoke about mutations. So we do have various mutations that occur as well. Within asexually reproducing organisms, we said that this is an identical gene transfer. So we're not getting that genetic diversity. How we are getting genetic diversity in prokaryotes is through horizontal gene transfer. Horizontal gene transfer. And so this is when we have genetic information from one organism going to another organism within the same generation. So vertical, being up and down, so it's going from one generation to the next generation. Horizontal is from one cell going to the other cell within the same generation. There's no going down. So this is how we allow for genetic diversity in prokaryotes, and we're going to take a closer look at that. And there are three ways that this has done that we are going to talk about, and that is transformation, transduction, and conjugation. So let's take a look at those three types of horizontal gene transfer that we see in prokaryotes. The first one is transformation. And in transformation, this is when the cell is going to take up DNA directly from the environment. And we've already seen this because we've talked about transformation when we talk about viruses, for example. But we see transformation occurring in bacterial cells as well. So transformation is when the cell is going to be surrounded by random bits of DNA, typically from cells that have been lysed, and then that DNA is going to be brought into that cell. So it's going to pick up that DNA directly from the environment. So we do see this happening a lot with say antibiotics, for example. So if we have a particular antibiotic that ends up lysing certain types of cells, but there are other cells in the body, other bacteria in the body that are, say, also pathogenic, then the bacterial cells that have been lysed due to the antibiotics may have DNA that's lying around. And that DNA that's lying around could then be picked up by the bacteria that is also in the area that's infecting the person, but wasn't affected by the antibiotic. So they can pick up that DNA, and this is one of the ways that we can get, excuse me, one of the ways that we can get antibiotic resistance, for example, is because if these cells are lysed, they may not be resistant to the particular antibiotic that the person just took that lysed those cells, but they may be resistant to some other antibiotic. And then those other cells in the area that were already resistant to the antibiotic that was taken can bring in those that DNA, those genes for antibiotic resistance to a different antibiotic. So then when the person has gone through that treatment with that antibiotic, they find out that they're still infected with something, and they take another antibiotic, it may not work because that cell has become resistant to that particular antibiotic, perhaps because of transformation, because of taking up DNA from its surroundings. Now, there are certain types of cells that can do this, and what this takes is competency. And competency is when a cell is going to attach to DNA naturally and then bring it into its cell. So a cell attaches to DNA naturally and takes it into the cell. So now typically double-stranded DNA is degraded by nucleases. So it doesn't typically bring in double-stranded DNA. However, if there's single-stranded DNA, so if it was broken down enough and there's single-stranded DNA around, this is typically not degraded. And since it is not degraded, then this can be taken up by other cells. So it's taken up and then it is recombined into the bacterial genome. And when this is done, then this would be considered recombinant DNA. So recombinant DNA is a molecule of DNA that contains fragments of other DNA. So this is a recombinant DNA, and then this cell would have recombinant DNA. So in this case, I mentioned that it may gain new phenotypic properties because of this. And so some of those phenotypic properties could be antibiotic resistance or antibacterial properties, or perhaps affect pathogenicity or virulence. So it could become more virulent now that it has this DNA that it just picked up, whatever genes happen to be in that. Or it could somehow affect its pathogenicity. So transformation can really shift and change an organism depending on what's picked up. So when transformation occurs, that cell, when it takes up the DNA, that could remain separate as a plasmid, or it can be incorporated into the host genome. So I can put that up here at the top here. So may remain separate as a plasmid. So remember, plasmids are those small circular DNA outside of the nucleoid region. So may remain separate as plasmid or incorporated into the genome. And it really just depends. It depends on the DNA, how big the DNA is, and what's going on with the cell already. So this is our first type of horizontal gene transfer for prokaryotic organisms. And so we see this transformation again between bacterial cells. So a bacterial cell will just have DNA outside of it. Sometimes they are plasmids, and so they'll bring the whole plasmid in. Sometimes it's just a random chunk of single-stranded DNA, and then it'll bring that into the cell. and then it is incorporated into the cell itself. The second type of horizontal gene transfer that we'll talk about is transduction. And this is when genes are transferred between cells in a virus. And so this one we have seen. We've already spoken about generalized transduction and specialized transduction. Remember, generalized transduction is any piece of chromosomal DNA transferred by accidental packaging of the degraded host chromosome into a phage head. So remember, this is when we have our bacteria and then it's been infected by a phage or a bacteriophage, and it has its information, then it's injected, and then when it goes in and it starts making copies of itself and it's putting the DNA in its phage head, it accidentally picks up some of the bacterial chromosome and gets it into one of the phage heads, and then when this bursts out, then it's going to be carrying that DNA with it. And so this is, again, generalized transduction, meaning any piece of DNA that happened to be in the region, because remember, when a bacteriophage goes into a bacterial cell, it lyses, or it doesn't lyse it, it cuts up, breaks apart the DNA so that it can take over on everything. So it goes in, it's going to accidentally pick up a chunk of DNA. Then we have specialized transduction. And this is where chromosomal DNA adjacent to the integration site of a lysogenic phage is transferred as a result of imprecise excision of the phage. So remember that we had our phage is going to infect the cell here. If we have our bacterial chromosome, then we have entered our viral chromosome, and then, so that's in the lysogenic stage, and then we go through the lytic stage and it cuts out its chromosome. When it does this, it accidentally cuts on either or both sides, and then when it repackages up this information, then it's going to leave with a chunk of bacterial DNA on either side or both sides. So this is our specialized transduction and our generalized transduction, which you should already be familiar with. So transduction is our second type of horizontal gene transfer. And again, why we're talking about this is because this is how we get genetic diversity. So as we are taking this newly formed virus here, now it's going to be taking all of this information and bringing it to the next bacterial cell, and then when it integrates that into the next bacterial cell, then it's going to have more information. It's going to be carrying with it more genetic information that's related to a bacterium, bring it into the new bacterium, and again, this could then give new phenotypic characteristics. So if it gives new phenotypic characteristics, then those things could be things like, pathogenicity or virulence or antibiotic resistant genes, things like that. So then this is kind of a hybrid of the viral DNA, and then the DNA from a previously infected bacterial cell is going to be brought into that new cell. The third type of horizontal gene transfer we're going to talk about is called conjugation. And conjugation utilizes some different parts of different bacterial cells. So the first one that you need to be aware of is the F plasmid. F stands for fertility factor. And so the F plasmid is going to actually code for the F pilus. So code for the F pilus. And what the F pilus is, and it also codes for some other things. Mainly the F pilus is what we're talking about in the F plasmid. So if a cell is F plus, so F plus cells, they contain the genes for the F pilus. These are also called donor cells. If it is an F minus cell, then it lacks the F pilus gene. Lacks the F pilus genes. And so then this is also called a recipient cell. So the F pilus is, again, it's for fertility factor, and that's where we have one cell, if it's an F plus cell, then it is considered the donor cell, and it has the ability to make this F pilus. This F pilus, the way this works, is it actually forms a pilus. Remember, a pilus is a projection of the cell. So let's say this is our bacterial chromosome over here. Then we have our plasmid. And that plasmid, this right here, is the F plasmid. And that's what makes this cell an F plus cell. So since it has the F plasmid, what it can do is it can make this pilus right here. When it makes this pilus, what it can do is when it senses that it is next to another bacterial cell, it'll actually make this bridge or this pilus. So then we have this other bacterial cell here, and then it has its chromosome, but it lacks the F pilus. So that's why this would be an F minus cell, because it doesn't have the plasmid here. So we have our donor cell and our recipient cell are F plus and F minus. And so then the one that contains the plasmid for the for the pilus makes the pilus, and the way that it works is it makes this projection. Once it makes the projection, then it's going to grab onto the other cell, and it's going to draw that other cell closer to it. When it draws it closer to it, then at that same time, the F plasmid here is going through rolling circle replication. So that cell or that plasmid is going through rolling circle replication. When it does this, it's making an additional copy, and when it does it, it's going directly through the F pilus into the other cell. As it does this, then it's kind of going around here, and then it's going to close up, giving a copy of the F plasmid, and then of course this will duplicate over here, and then this has already duplicated over here. So then it makes this F minus cell now an F plus cell. Now, this now gives this cell here the ability to make a pilus, and it can go on and it can make a pilus and then pull in another F minus cell and then pass along that plasmid. And the reason that that is important is because, one, this is how they transfer information here. This is kind of a bridge between the two. That bridge allows them to not only transfer that F plasmid between the bacterial cells, but it allows for other DNA to be exchanged through that F pilus. So not only because other things can be on that F plasmid, meaning it could have picked up DNA from something else, and if it does pick up DNA from something else, then it can bring it over into the other cell, but also because this bridge being open allows for exchange of information. If not on purpose, on accident, we can get something from one cell into the other cell. So we have our F plus cell, or F pilus, comes in contact with an F minus cell. It retracts, forming a cytoplasmic bridge, and then rolling circle replication happens of that F plasmid, and it's transferred to that F minus cell. So when this happens, we also have another cell that we can put a name to or a term to, and that's an HFR cell. An HFR cell, that stands for high frequency of replication. This is when the F plasmid has then integrated into the DNA chromosome. So it was an F plus cell in that it has that F plasmid, but then that F plasmid integrates itself into the chromosome. Once it has done this, it's integrated into the DNA chromosome, it now has the ability to, when it excises itself, accidentally grab DNA from either side of where it has placed itself inside of the chromosome where it's integrated. So something similar to specialized transduction occurs here, but it's not with a bacteriophage, it's just within the cell itself. So if we have our bacterial cell, and we have our host chromosome, and we have our F plasmid, then what we see is this F plasmid is going to be inserted or integrated into the chromosome. So then now that is part of the entire chromosome. Then when it goes to excise itself at some point, it imprecisely does this. And when it imprecisely does this, it's similar to, again, our specialized transduction, except this is just within the cell itself. So it's going to make the pilus, for example, with another bacterial cell. And then we have our chromosome here with the F plasmid in it, which this is now called the HFR cell. So over here we had a separate F plasmid. And so this was an F plus cell. So then once it has integrated into the chromosome, now this is an HFR cell, or high frequency of replication cell. Then when this cell is going to go through the process of excising this F plasmid again, it's going to kind of wrap over itself and cut. So what that means is it kind of does this twisting process here and will cut out the plasmid. When it cuts out the plasmid, then it may cut imprecisely. So my X should really be where that line is, but so it cuts imprecisely. Let me do a better job at that. So we're going to get rid of this. So when it does its cutting here, so it's going to go over a little bit here, and then we'll say it goes to here, so that when it breaks at this crossover point, you can see that there's some blue in here. That blue is then part of the original host chromosome rather than it being part of the F plasmid. So then when this, remember this has the pilus here and it's got another cell, then when this goes through rolling circle replication and goes into the other cell, it's going to bring with it whatever this contained here. So whatever genetic information that is. So then when that happens and this is going to be transferred, this is called an F prime cell, or an F prime plasmid, I'm sorry, F prime plasmid. So this plasmid here now is called an F prime plasmid because it not only contains the typical F plasmid material, genetic information, but now it also contains another piece of genetic information or another whole gene. So now that this F prime plasmid has been created, then it's going to be transferred to that F minus cell up here. When it's transferred to that F minus cell by conjugation through that plasmid being formed, then it can be maintained as an F prime plasmid, so it can stay in the new cell just as a plasmid, but it's an F prime plasmid because it has that additional genetic information, or it can be integrated into that host chromosome. And then, of course, that would then be called an HFR cell because the F plasmid is incorporated into the host chromosome, but then also it will likely be cut out and it will be excised imprecisely and carry with it more genes when it moves on to the next cell. And not only do we see this with the F prime plasmid, but we can see the HFR cell itself may be treating its entire chromosome as an F plasmid. So another thing that we see with these HFR cells is that they may treat the entire chromosome as an F plasmid. So recall that the genetic information for that plasmid is within the entire chromosome. So then when it does go through conjugation, if that plasmid hasn't been excised, then the cell might treat the entire thing as a plasmid. So it might take its time and try to go through rolling circle replication on the entire chromosome along with that F plasmid. So it tries to transfer the entire chromosome. Tries to transfer the entire chromosome. But the contact between the two cells is temporary. So contact between cells is temporary. So the genes that are closest to that integration site where the F prime or where that F plasmid was integrated in the chromosome is transferred more quickly than genes that are far away from the integration site. So we can actually take a look at this and we can have this process occurring and we can use this process in a laboratory to determine where genes are located on bacterial chromosomes. So we can see where the F plasmid is integrated into a cell. Then we can take those HFR cells and then have them be conjugated or go through conjugation with an F minus cell, and then we can see once we go through the process of taking a look at the cells that have been produced, we can actually see how close to that integration site different genes are. And so then we can actually map out the bacterial genome utilizing conjugation. So if we take a look at what this actually looks like in a drawing here, we have our bacterial cell, and then it has its chromosome, and it has the F plasmid integrated into that chromosome. So then we can say that we have different genes around here. So gene 1, gene 2, gene 3, and gene 4. So then we have our integrated F plasmid, we have our HFR cell, and then it's going to go through the process of conjugation, and it's going to treat its chromosome like an entire plasmid. So then when it goes through rolling circle replication, then it's going to start within that F plasmid. So we can say that this is, we can have both sides here so that we can imagine the rolling circle replication. So in rolling circle replication, remember that it goes around and it kind of peels off this outer layer. So if it's going to start within our F plasmid here, then it's going to kind of take this entire piece, it'll start to go around, and as it does this, this is actually peeling off here. So we can imagine that we have cut this here, and that this piece now is going to be going around, and it's going to be going through our pilus. So this little chunk of the F plasmid is start to go in there first, but since it's not just going around a plasmid itself, it's going around the entire chromosome, it's going to go all the way along here, and then we can map out. We can say gene number 1 is closest to the integration site, then gene number 2, then gene number 3, gene number 4, because we'll get various cells that have various pieces of the gene. So one cell might get from here to here before the pilus breaks and conjugation stops, or the second one might get from here all the way to here. And so we have lots and lots and lots of cells that'll have gene 1, for example. And then we'll have lots of cells that have gene 2, but not as many as gene 1. And then last cells that have 3, and then very few cells that have 4, for example. And then very, very few cells that have the entire F plasmid. And so then we know that it's gone all the way around, and it has genes 1 through 4, and then we know the genome of that particular organism, which of course 1 through 4 is very simplified, but hopefully you get the idea there, is that we can then figure out what's gone into the other cells and utilize that information to map a genome. So there are other plasmids, so just a note about plasmids in general, is it's not only the F plasmid that gets moved through conjugation. So other plasmids can be moved through conjugation as well. So transferred through conjugation. So it is the F plasmid that has to initiate that conjugation, because it's what is actually going to encode for that F pilus. But then again, once that F pilus is open, once that bridge has been made, then things can just move through. And so other plasmids can be going through rolling circle replication at the same time, and as they are, they're moving their plasmid into the other cell. So we see some things like toxin-producing genes that are able to move. Toxin-producing genes are transferred on plasmids. We also see different metabolic enzymes. And when we see metabolic enzymes, that means that we are going to have metabolic changes. So maybe something that could only exist on utilizing glucose for its carbon source, for example, then maybe we'll start to be able to use lactose. And so now that brings a whole big change to this particular organism. Maybe it can live better in different parts of the body, for example, and cause more infection, or live longer outside of the body before it infects somebody else. So if we have these different metabolic changes, that can come from conjugation and moving over those plasmids. Then we also have R plasmids. And R plasmids are specific to antibiotic resistance, and that's what these are, what these stand for. The R plasmids are plasmids that contain genes that code for antibiotic resistance. And oftentimes we see that these R plasmids contain genes for resistance to multiple antibiotics. So we see through these three different forms of horizontal gene transfer, through our transformation, our transduction, and conjugation, we can actually end up with plasmids that contain multiple resistances. They also contain genes that control for conjugation and transfer of the plasmid. So not only do these plasmids, can they contain multiple genes for multiple antibiotic resistances, are resistant to multiple antibiotics, then they also contain that conjugation ability. So these plasmids kind of act as their own entity here, where they can actually make the cell have a pilus and then transfer this plasmid over to a new cell, which then can make lots of resistance to lots of different antibiotics in lots of different cells. So this can happen within its own species, so within its own generation or multiple generations of its own species, but this can also happen between species as well. So again, if you are infected with two different types of bacteria, they're near each other, we may see some of this movement, including conjugation and moving of these R plasmids. So the last thing that we're going to talk about in this section are called transposons. And a transposon is a molecule of DNA with inverted repeats at the ends that also encode for the enzyme transposase. So if we take a look at what we're talking about here. So we have the DNA, which keeps going this way, and the DNA, which keeps going this way. We're going to put a gene in here, and then also we're going to put in our transposon. So our transposon has the enzyme in here. This is the enzyme for transposase. Transposase. And then on both ends, we have these inverted repeat regions here. So these inverted repeat sequences actually are going to kind of match up with each other. So as the cell is going along, it's transcribing and translating. It's going to translate this transposase enzyme. The transposase enzyme then can come along and grab these two sections here, kind of cross them over like we saw earlier with the F plasmid excising, cross them over and then cut out this section here. Now, a lot of times what happens is it can cut out this section here, but then the enzyme also is going to interact with this gene over here. And what it can do is it can allow movement from one location to another. So it's transposition. So it's moving positions. Trans is movement, and then position. So our transposition, and that is found in both prokaryotes and eukaryotes. I'm actually going to bring in the picture here from the text so we can see how that folds over. All right, so here is the image from the text with our transposon, and then you can see what I had drawn here previously, where we have that inverted repeat, and then we have the transposase, and then as it's being transcribed and translated, we actually end up making transposase here, and then it's going to go in and grab this chunk of DNA, and then it's going to move things around because it interacts with this gene as well. And so it's going to move itself, and it basically does this on its own. The transposase goes in and it moves this chunk of DNA that codes for itself around the genome. So what we see is that it is going to relocate genes, it'll disrupt genes, and so when it disrupts genes or relocates genes, then it's going to change the phenotype. And again, why it's important that it can inactivate or activate a gene is because it can alter this phenotype. And in ways that it can alter the phenotype is they can make more virulence factors, so it can adjust the virulence of the bacteria, or because it can confer antibiotic resistance. So then it can move it from the chromosome to a plasmid, for example. So if there are genes on the chromosome and then this transposon is in the chromosome, it can then take those genes and move them to a plasmid. And then if it's moved to the plasmid, then they can be transferred to other bacteria through one of the horizontal gene transfer techniques or methods that we saw. So this is just another way to get genetic diversity in prokaryotes. The transposon is kind of moving itself around just kind of randomly, and then it's going to change this phenotype, change virulence factors, change antibiotic resistance, and so are the other three real ways that we see happening more often. So transposons are kind of random and not found in all cells, but we do see transformation, transduction, and conjugation happening throughout bacteria.